access to the brain and neurons and neurocognitive aspects. So if we think about what are the factors which determine food choice, we and other people have mapped out multiple aspects of that. For example, right, reproductive state in women and also insects and rodents impacts how much and what type of food, especially amino acids and salts, women but also animals will eat. Another important aspect is the metabolic need of specific organs. So organs speak with the brain and through hormones and peptides instruct what they need and will change the food intake and food choice of animals. But one thing I want to emphasize today, and I'm going to discuss a bit and focus today, is the impact of the microbiome. So I think there is an increasing awareness that our gut microbes have a strong impact on brain function and also neuronal pathologies, right? But we are really at the beginning of understanding what are the mechanisms by which actually gut microbes impact brain function. I think one very attractive hypothesis which I would like to highlight in this audience, because that's a bit the theme of today, is the idea that microbes in our gut obviously interact with the diet and nutritional components in the diet and modify them and manipulate them. And if you think that, or if you believe that nutrients have a strong impact on the brain, then obviously this interaction of gut microbes with nutrients, including the production of nutrients by the gut microbes, will then act on the brain. And so this is an interesting and I think very attractive hypothesis to think mechanistically about how gut microbes could affect brain function. But there are many, many open questions which I think we need to tackle as a field, right? First of all, how do nutrients shape the microbiome? Why do we need communities? Because I think one thing which has emerged also is that it's not single bacteria which affect physiology, but it's communities. Why do we need bacterial communities? And then just very, very importantly, obviously, what are the mechanisms by which gut bacteria change behavior? And I think there Drosophila can be a very attractive model. First of all, Drosophila has a very conserved genome, so molecularly Drosophila is very similar to rodents, vertebrates, and also humans. And here especially metabolic pathways and regulatory systems like insulin, leptin, etc. are conserved. But also the organization of the physiological systems like organs are also conserved, right? So the digestive, the body organization, the nervous system, including also metabolically active organs, are very similarly organized and function between humans and insects. And I'm sorry that I cannot move the pointer here to highlight some things. And I think the power of Drosophila is also highlighted by the multiple Nobel Prizes which have been given over the last 100 years to work which has been done in flies. And here I would also like to highlight the last Nobel Prizes which are related to neuroscience, the discovery of chemosensory systems, innate immunity, and also circadian rhythms, which all have been shown to be conserved from flies to humans. But what do we know about the microbiome in flies? Well, flies have a very simple microbiome. If you catch Drosophila in your kitchen, you're going to find mostly 5 types of bacteria. Lactobacilli and Acetobacter are going to be mostly prominent, similarly than we also find in vertebrates. And they have been shown to have a lot of different impacts throughout the whole lifespan of flies, from development, where they're very, very important to promote growth, to adults, where they change lifespan, tissue homeostasis, and feeding preference. And interestingly, the same bacteria can have the same impact on vertebrates. So if you take the Lactobacillus plantarum, which promotes growth in Drosophila, and you put it into a mouse, which has been kept in malnutrition conditions, you're going to be able to rescue the malnutrition effects on growth in mouse by putting in the same bacterium which has been isolated in flies, suggesting that there's a functional conservation, right? So, but we want to understand food choice. So the way we measure food choice in flies is using the fly pad. This is the same principle as you use on your iPhone and iPad if you're bored and now are reading your emails instead of listening to me. And what it does is it measures the touch of the fly with food, and from that we can measure every single food intake event from the fly. So what is it we want to really understand, right? So if you look at 3 different types of flies, flies which have been kept on a full diet, flies which have been kept on an amino acid deficient diet, and flies which have been kept on a carbohydrate-deficient diet, and you give them the option to eat from amino acids or from carbohydrates. So this is like a buffet choice option. Then what you're going to see is that flies which have been kept on an amino acid-deficient diet, they will specifically increase the intake from amino acids, but not from carbohydrates, whereas carbohydrate-deprived flies will only increase the intake from carbohydrates and not amino acids and proteins. So they're able to identify a lack of specific nutrients and then compensate by eating from that nutrient. And humans do that too. So if you take people and put them on a low-protein diet for 2 weeks and then give them the option to eat whatever they want, you will see that they will overeat proteins, most likely to compensate for the lack of proteins. So the brain is able to solve this problem, right, to eat right. It's able to detect that it's lacking specific amino acids or nutrients and then compensate by choosing them from the diet. And I think this is very, very at the basis of understanding nutrition and how the brain controls nutrition. But let's get back to the microbiome, okay? So this is a classic experiment we would do with our flies in the lab. So you have flies kept on a full holytic medium. So this is a fully chemically defined medium in which we can manipulate every single nutrient. And we're comparing flies kept on a holytic medium, which has all amino acids, to a fly which has been deprived of only one essential amino acid, which is isoleucine. And what you see is that there's a drastic increase in protein intake. Yeast is the main protein source for flies when they're isoleucine deprived. Now this is seen mainly in flies which don't have a microbiome, so in germ-free animals. If we put back 2 specific bacteria which are found in wild flies, Acetobacter pomorum and Lactobacillus plantarum, now these flies, even if deprived from isoleucine, will not be eating protein. And you need both bacteria because only putting in Acetobacter pomorum or only putting in Lactobacillus plantarum does not suppress protein appetite. So these 2 bacteria have the ability to suppress protein appetite in the flies. So the question now, how do gut bacteria change food choice, and why do you need a community of 2 bacteria to change behavior? And here what we have been using is isotope-resolved metabolomics. So isotope-resolved metabolomics allows us to trace in vivo in the fly or ex vivo in culture the route by which specific metabolites which are marked with non-radioactive heavy isotopes are modified by the bacteria or by the animal. And so what we can show, and it's too bad I cannot point here because it's a bit of a complex slide. So what you're seeing here is the amount of isoleucine being produced in culture in conditions where you have either only Acetobacter pomorum or Lactobacillus plantarum, and we are only looking at the amino acids produced from labeled glucose. And what you can see is that you need both bacteria to be present to produce significant amount of isoleucine in culture, and we have also new data showing that the same thing is in vivo. And we can trace that back to lactate, and so what happens is that lactate is being produced by lactobacillus, and so if you put labeled lactate alone together with Acetobacter pomorum, you can see that now these bacteria produce high amounts of isoleucine on their own using the lactate we put from the medium. So does this matter in vivo, right? And so another thing we can do in flies is we can do bacterial genetics in the animal. So what we can do is we can mutate the ability of lactobacillus to produce lactate and check if this is important for changing behavior. So again, here, the control experiments. If you take flies, remove isoleucine, they increase protein intake. Acetobacter pomorum alone doesn't do anything. Acetobacter pomorum with lactobacillus now suppresses protein appetite. And if we mutate the ability of lactobacillus to make lactate, we cannot get a rescue. That's the second last bar there. And we can rescue that by putting back lactate in this mutant bacterial background. So the question is Acetobacter and lactate sufficient to change behavior? Again, we can take now a condition in which we have Acetobacter alone, which doesn't suppress protein appetite. And if we add lactate, lactate together with Acetobacter pomorum is sufficient to completely suppress protein appetite in these animals, and that's not because lactate has specific effects, because if you put lactate alone in there, there's no effect. So lactate is a key bacterial metabolite being produced by lactobacillus, which is required to change behavior, and Acetobacter is the key bacterium altering behavior. Now, I won't show the data, but it turns out that this is really important because the reason the lactobacillus need to produce lactate is so that Acetobacter pomorum can make amino acids, and this allows lactobacillus to now survive in the gut of the host because lactobacillus cannot make isoleucine. Okay, so that means that there is a circular nutritional economy there. And this metabolic cross-feeding allows the gut bacteria communities to remain stable even if important nutrients are missing. But the amino acids are not what changes behavior. And if you want to know why we get to the conclusion, you need to read the paper. I don't have time today. Furthermore, this also shows us that Acetobacter is the key bacterium which changes behavior, and that lactate is a precursor to produce psychoactive substances. And I think this highlights the power of having complete control over the diet and the use of isotope-resolved metabolomics to now look what are the metabolites produced by Acetobacter from lactate, and that's something we're doing now. And to understand what happens in the brain, we can do whole brain calcium imaging in the fly. This is what you're seeing here. So this is a fly brain now with GCAMP. And we can see the fly brain neurons, all the neurons in the fly brain, how they are activated when the fly is eating. So we can watch the brain of the fly while it's eating. And we can do that in flies which have or don't have a microbiome, or flies in which we reconstitute the metabolome of the fly by putting in specific metabolites. So I propose here that because nutrients have such a strong impact on behavior, that by looking at neuronal circuit function, molecular mechanisms involved in nutrient sensing in the context of nutrients and the microbiome, using quantitative behavioral assays, putting that in the context of phenotypes like aging and health, and taking into account metabolism and organ physiology, there at the center is really where we can start to understand mechanistically how nutrients act on the brain and how body-brain interactions shape brain function. And obviously that's best done in flies. And this is not only just for food intake, because if you compare flies which are fully fed with protein-deprived flies and look at how they search for food, you see that they do use very, very, very different strategies when exploring the environment and looking for food. Again, showing that the nutrient state of the animal, and here we're talking specifically about the protein state, affects cognitive-like behaviors, which are obviously related to foraging and therefore are involve complex cognitive computations. These are the fantastic people who have done the work. If you're interested in working with us, would be the view from your working place. And I invite you to watch our talks. We have every Monday a seminar series, a virtual one on brain-body interactions, in which we discuss these type of phenomena, which we think is actually very relevant for all kinds of pathophysiological understandings of brain function. Thank you very much.
Thank you, Carlos. That was really fascinating. And now we are only missing the dessert. And the dessert is served by Giulia Chiacchierini. She's a postdoc at the Institute of Technology in Italy. And the talk is The Dessert, Neural and Behavioral Analysis of a Nutrient-Specific Appetite for Protein.
So we finally arrived at the final course of this tasty symposium. I would especially like to thank the organizing committee for having me today, giving me the opportunity to replace my PhD project supervisor, Jamie